welcome back to the course of plant developmental biology. So, in last class we were discussing reverse genetics based approaches which we are taking for to study the developmental. So, in that class we have covered identification and isolation of genes for developmental functions. Now, we started the functional genomics based approaches. So, in functional genomics based approaches, what we discussed, the first step was monitoring gene expression, and now so we have seen how to check the expression pattern of individual genes. Now, we will mostly focus on the global genes, how to study the expression pattern of the global genes and what are the way how to study the expression pattern. The important thing here is that you can again fix the problem. So, the main goal of genomics or global gene expression analysis is to identify the differential genes in different conditions. It can be different organs, it can be different tissues, it can be different stages, it can be different conditions between a wild type and mutants, any combination whatever you can choose with respect to the development, and then you take the genomics based approaches and identify all the genes which are differentially expressed in between two conditions, two organs and whatever. And there are many way to study differential gene expression. So, I will list some of them here, but we will focus on only few. So, these were way to study. First was the serial analysis of gene expression says. Here people started by obtaining some short sequence tags from a large number of cDNA clones, and then they were sequencing it and identifying the differential gene expression pattern. Then another way was differential display RT-PCR. Here what people used to do, they used to take a two population of total RNA, they used to make cDNA, they used to run a gel and then try to identify which particular band is missing in one condition and present in another condition, or vice versa, and then they used to cut that band, amplify the fragment, clone, sequence and identify what are the genes. Another way of doing was the subtractive hybridization. This was PCR based amplification of only cDNA fragment that differs between two conditions. And so, basically here what you used to do, there were two population of the messenger RNA, you make cDNA, then do the subtraction. After subtraction you identify or you isolate only those populations which are differentially expressed, and then specifically you can amplify them and then identify them. Then the this technique was very widely used in most of the genomics based expression differential expression pattern analysis, which was microarray. Microarray was the technique where small fragment or small oligo single stranded oligos were spotted on a chip, and then the total RNA maybe was isolated from a particular condition to identify the differential expression. We will see microarray in great detail how this has been used, and this is one of the most used technology in the plant developmental biology. And now recently next generation sequencing has actually changed the entire scenario. Now we are doing a lot of RNA sequencing, and this has lot of benefits over the microarray. So, these are the different methods which are being used to study the differential expression pattern in different conditions or different developmental processes. So, before going to the microarray and RNA sequencing in detail, I would give some of the typical examples of different technologies which was used. For example, if you look this research here, the transcript profiling was done in rice seedling using SAGE as a method. So, this was used and people have identified the differential expression here. Then this is one example where differential display RT-PCR was done. So, this is MATS box containing transcription factor in rice, and what was important here that this gene which is MATS1, it was expressed in sepal equivalent, which in case of rice is lemma and palea, but it was not expressed in the inner organ. But when the gene was mutated, when this gene was silenced, the effect was also seen in the inner organs, inner floral organs, for example, lodicules, stamens. Then this was the question that if the gene is not present in this tissue, how it is regulating. So, one of the possibility was that maybe it is activating because at very early stage it was expressed throughout the floral primordia. So, it was hypothesized that maybe it is activating some early regulators at very early stage whose expression is basically there in inner floral organs and regulating it. To identify such kind of gene, the differential display RT-PCR was taken up, and then one of such genes was identified, which was OSMGH3, and it was shown that actually OSMGH3 was expressed in the floral, inner floral organs. So, this is again, if you look, this is northern blot, and if you look the expression pattern of OSMGH3, you can see that in wild type leaf sheath it is not expressed, whereas in the young panicle, panicle is inflorescence in case of rice, it has a very high level of expression. But if you look a panicle which is mutant panicle where MATS1 is down regulated, the expression of OSMGH3 is also disappears, which means that first thing that there is a organ specific or tissue specific regulation of this gene. The gene is only expressed in the panicle, not in the leaf sheath, and its expression or its activation is dependent on the MATS1. So, this is one example of differential display RT-PCR. Then subtractive hybridization was used in this study where they have done a comparative transcript profiling of gene expression between seedless variety and its CD wild type during floral organ development, and I will not go in detail. Then the technique which I was going to talk about is microarray. So, as I said that microarray is a kind of chip. So, this kind of chip was generated, and this was very widely used in the post genomic era before RNA sequencing. Once the genome was sequenced, we identified the genes, we identified the sequence of all the genes, then those genes, the probes for those genes were designed, and these probes were spotted in these chips. So, for every gene we had a kind of probe, and then there was two way of identify the differential expression. One was the two color way and one color way. So, in one color way what you have to do, let us assume that you want to test a wild type sample which is your test, or mutant sample. So, you want to identify the genes which are differentially expressed between this wild type and this mutant. Then what you can do, you can extract total RNA from the wild type, total RNA from the mutants, then you label this RNA, and one way of labeling was the biotin labeled cRNA synthesis. Both were basically labeled. So, entire RNA population in these two conditions were labeled, and then these labeled cRNA were used to do the hybridization in the chips. So, now these chips contains global genes, all the genes represented in an equal amount. Now, since let us assume that there is a gene A which is more in case of wild type, but it is getting down regulated in case of mutant, then you are expecting more cRNA in wild type, less cRNA in mutant, and when you hybridize on the chip, you are expecting more signal for that probe in case of wild type, whereas less signal in case of mutant. And then if you compare the signal intensity, you do the data acquisition, scanning, and if you calculate the absolute value of hybridization, you can identify the difference in the expression pattern of gene A between wild type as well as mutant. Then another way of analysis was that, let us assume the similar case, you have wild type RNA, you have mutant RNA, and then you take total RNA from both the sample, and then you label them with two different dye. For example, this is Psi5, this is Psi3. Psi5 will give red color, Psi3 will give green color, and then you take equal amount of total cRNA and hybridize on the same chip. So, basically the difference between these two methods was, here both the cRNAs were labeled with the same label, whereas they are labeled with the different labels. Here they were they were hybridized on two different independent chips. Here both are being hybridized on the same chip. So let's assume if there is a chip which is for gene A. Now normally if the expression level of A is same in wild type and mutant, then you are expecting same amount of Psi5 label cRNA and Psi3 label cRNA to go and hybridize with this particular probe. So you are expecting same amount of red signal, same amount of green signal, and the result will be yellow signal here. But if any one of them are differentially present, for example, if expression is more in the wild type, then you are expecting more red signal than the green, and then the color of these spots will change. It will be more towards the red. If gene is down regulated, then it will be more towards the green. So based on this color and then the quantification, you can actually calculate the fold change between wild type and mutant for all these kinds. These techniques you can also use for the different stages. It's not only in wild type and mutant. For example, if you take this particular case, so where what people have studied the genes which are differentially expressed at different developmental stage of rice panicle development or flower development. So for example, if you take these stages, this is stage 0, which is basically vegetative phase, and this is your shoot apical meristem. Extract total RNA from vegetative phase, and then this is a transition from vegetative to the reproductive phase. Then after transition, this inflorescence is undergoing a different stage of development. Here primary branch meristem, and then secondary branch meristem, secondary spikelet meristem, all these developmental stages it is going across. So what people have done, they have taken RNA from here, from all these different stages, and they have performed the microarray analysis in great detail, and this how they could actually identify several things. First thing, they could identify the gene which is present in a particular developmental stage but absent in the different developmental stage, or a particular genes how its dynamic expression pattern is across the development. For example, some genes might be 0 here, and then they are suddenly getting activated and they are maintained across the development, or they are activated in this developmental domain and then they are going down. So this is a basically dynamic expression pattern you can analyze using this technology, and this everything we are doing at the global level, and we can identify. For example, if some of the genes they has been tested here, so you can check this is RT-PCR analysis. If you see the gene LEX, the gene LEX is very low at 0 stage, but across the stage is 1, 2, 3, 4, 5, the expression level is increasing. Whereas if you take this FGP, which is frizzy panicle, this is the expression starts or activates only from the stage 4 onwards and continue in the stage 5. It is not present in the early stage, whereas this MATS3 it is getting activated in stage 5. So this is how the microarray was extremely useful in studying this kind of genome wide differential expression analysis. This is another example of microarray which was used, and here you can look here that they have focused their study on the MATS box containing genes. Why I am focusing most on the MATS box containing gene, because this was the class which is well established to regulate lot of developmental processes. They are very, very important with respect to the plant development. And here they have taken large number of tissues, mature leaf, roots, 7-day-old seedling, and then this panicle, different size of panicle or inflorescence signifies different developmental stages in case of this one. And then they have checked the expression pattern through microarray analysis, and what they can identify, there are a lot of genes. So if you look, the expression is here. So if green is less expression, red is high expression, and then you can look that a particular gene, if you grow across the different organs, you can see their expression, how the expression patterns are changing from one developmental stage to another developmental stage, or one tissue to another tissue. So, this was important study. That now nowadays it is RNA sequencing. This is next generation sequencing, is very cheap and very easily available. So, now people are doing RNA sequencing, and RNA sequencing has some advantages over the microarray. First advantage is that for microarray to generate the chips, you have to have the sequence information, which means that the genomes of the organism has to be sequenced. Then and only then you can define probes for the chip. But in RNA sequencing, the genome, even the genome is not sequenced, it can be studied through RNA sequencing. Second important thing, one very important process in eukaryotic organism is alternative RNA splicing. Alternative RNA splicing is like if a gene has multiple introns. So, let us assume this is intron 1, intron 2, intron 3. So, there are a large number of gene regulation occurs at the level of splicing. So, alternative splicing essentially provides an opportunity to generate more than one type of protein from a single gene just by alternatively splicing the different introns. For example, in one messenger RNA you can generate from here, if you splice all the introns, then you can generate a messenger RNA where you do not splice intron 1, or you do not splice intron 2, or you do not splice intron 3, or you splice individually, or you can even skip the introns, you can splice this exon along with the intron. So, this provides a large number of different transcripts from the same gene, and it is very difficult, or it is very challenging to keep probes for all splice variant in the chip in the microarray experiment. So, it was very challenging, because if you do not know how many splice variant exist for a particular gene, it is very difficult, because normally when you are designing the chip, you used to keep 1 or maybe 2 or maybe 3 chips for a particular 3 oligos for a particular gene. But if there is some intron here, you will not have intron or splice variant specific chips. But in case of RNA sequencing, basically it can capture all the splice variants. So, if you look the technique how RNA sequencing functions. So, basically what we do in the RNA sequencing, you extract total messenger RNA, and then you can specifically remove messenger RNA from the total RNA. How you can do that? You all know that messenger RNA, they have poly A tail at the 3' end, and then you can use oligo DT tagged with some kind of bits, and using this system you can specifically remove messenger RNA from the total RNA population, and then you use this RNA and you can do the fractionation. You can make a small fragment of RNA and then use some random primers. Random primers are basically small 6 nucleotide long single nucleotide primer with different sequences. So, it can randomly go and bind different places, and then you can make first strand cDNA synthesis, second strand cDNA synthesis. So, basically you are generating a different small fragments cDNAs, double stranded cDNAs. And then one important thing what you can do, you can put the phosphate. Normally the primers, they do not have phosphate at their end. So, then you can provide phosphate, because if you have to use this for further ligation process, if they do not have phosphate at their end, then they cannot make a phosphodiester bond. So, you can do the end repair, put the phosphate, and another thing what you can do, you can add flanking A. This you can do using poly A polymerase, and then if you add this A tail, this will provide you to take this fragment and add some adapter. So, you can ligate adapter. This adapter can have a stretch of T. So, T and A, they will go and they will anneal to each other, and then you can perform a ligation reaction. So, essentially what you are generating double stranded DNA fragments with adapter at both the end. and then you know the sequence of this adapter, you can use this adapter sequence and you can take and sequence this entire cDNA population. This is very widely used these days, and this can be used to identify. So, if you take two different population of RNA, two different population of, then you can generate two different population of fragmented cDNA, and if you sequence them and compare them, you can identify differential expression level of different RNAs. So, again I will give some of the examples where RNA sequencing was used to identify differential expression pattern. For example, if you take this, so here different stage fruits were taken and total RNAs were extracted and RNA sequencing was done, and you can clearly see different genes and their expression pattern. For example, if you look here, so if you look this gene or any of these gene, this has a very low expression at this stage, but if you come here by the end of this stage, the expression level is really going very high. So, this tells that there are a differential expression of different type of genes, and here you can see that this is a heat map of RNA sequencing data which involves the shoot development. So, they have taken different types of shoots here, secondary shoots, primary shoots, then there is something called mother bearing shoots, and they have extracted total RNA and they have performed RNA sequencing across these different types of shoots, and they have identified different tissue specific genes which are present here. This is another example where comparative RNA sequencing analysis was done for transcriptome dynamics during petal development in rose, one of the rosaceous member, and where you can see that they have taken different stages of the petals from the different developmental stage, and they have performed the RNA sequencing analysis, and they could identify a different genome expression pattern. And you can see they can identify here all these different types of transcription factor. Depending on your study, you can focus on transcription factor, so you can identify all the transcription factors which are differentially expressed or anything, any other kind of transcription factor. Another way, if you take this kind of RNA sequencing, and RNA sequencing you can combine with a different other techniques. So, it is not standalone technique, and then you can perform a more specified or more defined way to generate RNA sequencing data. For example, if you have some kind of developmental stages, maybe I will draw here. So, for example, if you want to study a developmental stage at a particular, let us assume that you want to study root developmental stage, and you know that root development occurs through different stages of the development. The first stage in the lateral root branching is that the specification of some cells. So, these are the mature cells. Now, they are taking stem cell property, and then these cells start the cell division. They undergo the process of cell division and they generate some kind of primordia which is called root primordia, and then this root primordia undergo the process of cell differentiation. It start root specific differentiation program, and then finally the primordia is emerging out. Now, if you want to really identify the genes, you can collect this tissue very specifically. There are a lot of techniques available. One of the technique which is called laser capture microdissection you can use, and you can collect all these primordia and then extract total RNA from these primordia, and then you can identify. Or you can take some wild type sample and the mutant sample when a particular transcription factor, a particular gene is missing, and you want to identify what are the genes which are whose expressions are getting affected when you mutate a particular gene. Then you can take total RNA, you can do tissue specific total RNA extraction, and then perform RNA sequencing to identify the differential genes which are present in these two particular conditions or two particular mutants. So, this is analyzing expression pattern, where we have tried to study today how to identify differential expression pattern. Now, for doing spatial and temporal expression pattern analysis, there are several methods which we will discuss more in the next class, but to just briefly introduce them, one way of doing is that to study RNA localization, which we are going to do by RNA in situ hybridization. Then you can do protein localization, you can use some specific antibody and localize, you can study their specific localization across the tissues, and then you can do a kind of promoter trapping where you can use a kind of promoter-less reporter gene, and you can identify if a promoter, or you can do transcriptional fusion, you can do translational fusion. So, maybe we will stop this class here, and in next class we will discuss in detail how to study spatial and temporal expression pattern and functional characterization of gene through reverse genetics based approaches. Thank you very much.